mark on its own. It needs to tell me why it's not valid, and then I needed you to link it. Okay, going on to section C, here is our theory questions. What I was looking for at this first one, talking about chloroform, was I was looking for simple diffusion or something about the word diffusion. If it didn't have the word diffusion, I couldn't give you that mark. Great with identifying the different enzymes. So which enzyme graph would have the bacteria in hot springs? That's definitely going to be B, which is this one up here. And that's because we've got much hotter temperatures that it's comfortable with. Now, the next question, some people seem to get a little bit confused. You're saying why enzyme B wouldn't function well in a human. So enzyme B isn't going to function well in a human because here is human temperature. So that's 1 mark for identifying human temperature of around 37 degrees, or the fact that humans' body temperature is much lower than this enzyme. And so therefore, if we're down here, having a look at this graph up here, if I was looking at enzyme B, we're really far down below that curve. Therefore, that means it's like it's too cold for the enzyme. If it's too cold for the enzyme, then therefore we get not enough energy for collisions or fewer collisions or something about collisions, not enough energy. That was your second mark. So 1 mark for identifying body temperature in a human and 1 mark for identifying fewer collisions. Down to question 9. Here's a substrate concentration graph. You needed to explain what was happening up here at the plateau. So 1 mark for saying all active sites are occupied. So if you're looking through this mark scheme and you see that things are underlined, that means that they have to be there for that mark. So it was not sufficient to say that all enzymes were occupied. So you could not say all enzymes occupied because it's not specific enough. You have to mention the active sites. Then you could have given me one of 2 kind of explanations. You could have said that the rate of reaction is happening at a constant or consistent or continuous or something about that being a maximum rate despite increasing the substrate concentration. Or you could have said enzyme concentration is now limiting. Moving down to question 10. I really liked this question talking about the blanching of corn. So when you think about blanching, we're talking about boiling water. So that means it's too hot. When we're talking about too hot, then and enzymes, we know that we always must include denaturation. So if you didn't say the word denaturation, and it is denaturation, not denaturization, then I can't give you 3 marks. So it didn't matter how many of these statements you had, these ones down here. If you didn't say denaturation, you couldn't get the 3rd one. So if you said denaturation or denatures, then you say something about changing in shape of the active site, substrate no longer binds, or changing in shape of the active site, breaking the bonds in the tertiary structure. I really wanted to see this last one, but I was happy to accept any 2 of those statements. So if the sugar is no longer becoming starch, the corn will stay sweet. That's linking it back to the context of the question. Okay, down to question 11. Question 11, the sequence of amino acids. What determines the sequence? It is not sufficient to write DNA or mRNA, okay? Because it wasn't saying what codes for amino acids in general. It was asking you about the sequence. And this one here, not many people managed to get it right. I needed the word sequence. So this is one of those hard lessons that we learn when we don't necessarily write that one word and you think I'm being really mean, but by getting it wrong now, you won't get it wrong the next time it comes around. Okay. Then we had functional and structural proteins. I was very generous on this question because I, in particular, had told my class you don't really need to remember their functions. If you could give me some generalized names, I would ignore the function part because I thought it was a little bit too far off the study design. So I was quite generous there. If you think that I have been too harsh on you, please let me know. Down to question 12, talking about the bacteria treated with malinate. So malinate is this substance here. It looks very similar to succinate, but it's binding to the active site. So it is a competitive inhibitor. So if malinate is a competitive inhibitor, that's 1 mark, okay? 1 mark for saying that it's the competitive inhibitor. Then you needed to tell me why increasing the concentration of succinate. So I didn't need you to tell me anything else about competitive inhibition. I just wanted to know why, if we put more and more succinate in, did the bacteria survive? And it needed to be something here, this statement here, needed to be a comparative statement. So you needed to say that there were more likely to be more collisions, or the succinate is more likely to come in contact with the active site. If that happens, then you're going to get more fumarate forming, and more fumarate means you can have more biochemical pathways or more energy, for the biochemical pathways. So it needed to have a comparative statement there. Next one, pretty good, DNA versus RNA. Just be careful on your spelling. Last year's exam, they didn't accept this one because it was actually too close to a... they didn't accept that, thiamine, because it was too close to a different concept, not necessarily thiamine here. If you can't remember how to spell them, guys, just use the letters. They accept it on the exam, so we'll accept it here. So you can just say T or you can just say U. The other thing I want you to be very careful of when you're talking about no oxygen, it's not that deoxyribose has no oxygen, it's that it has less oxygen in the sugar, and the ribose has more oxygen in the sugar. Sorry, let me just highlight that. It's less oxygen in the sugar. So if you said it had no oxygen, I couldn't give you the mark. All right. I ended up breaking this one down a little bit, even though it was out of 2. So what you were looking for with your mRNA is that it was complementary and U instead of T. Yeah, and then you used this sequence to read off this table. So if I do this first codon here, U, I go the first position is a U, the second position is a U, and the third position is a U. So that's this one here. So that's why it's phenylalanine. I'm just going to do the second one. So CAC becomes CAC. So where do they all meet? It's this one here, histidine. So you can see that's why it became histidine, and that's why this one's phenylalanine. Okay. So just moving on to the next question, because I want to spend a little bit of time here, we can see that if AAA mutated and became AAG, its codon would then be UUC. This one's going to be CAC still. This one here is going to be U. And the last one is going to be CCC. So this one here was very poorly answered, but it was meant to be. It was quite a tricky question. So you can see here is your new mRNA sequence. UUC still codes for phenylalanine. So UUC still codes for phenylalanine. CAC still codes for histidine. GUU still codes for valine. But the final codon that's changed, we now have a different amino acid. So we've changed the amino acid from serine to pro. And you could have done that up here. You could have done that somewhere else. I looked at the whole question to see whether or not you could show me that that last amino acid had changed. So that was 1 mark for identifying that the last amino acid had changed. Now, you could, without doing that, have got the 2nd mark. The 2nd mark was for telling me that the primary structure or the sequence of amino acids has changed. Now, because it's changed, therefore your protein is not likely to function. Due to change, protein, you could say protein won't fold correctly or might not fold correctly. There's a whole bunch of different ways you could say this, but essentially that's what I was looking for. And the very last question, I was really impressed with what you girls did. Lots of people managed to get at least 2, if not 3 out of 3, if you were on the right thing, on the right track. It says describe the process by which insulin can be manufactured and leave the pancreatic cells. Now, a few of you focused on the steps in protein synthesis, and then you went on to tell me these steps here, but I ignored them. So I ignored anything that you wrote about protein synthesis, because otherwise this question would have been worth about 6 marks instead of 3. So what I was looking for were the words that are underlined here. So you can see that insulin was going to be manufactured in the ribosomes on the surface of the RER. It then was modified by the RER. Then the Golgi did packaging and modification. And what I was looking for was the vesicle fusing and releasing the contents via exocytosis. So what you needed here was any 3 of the following. So any 3 out of 4. And so that's the mark scheme. And I'm happy to answer any individual questions that you girls may have. Thank you.